GABA is an amino acid that is an important inhibitory neurotransmitter which is found in the central nervous system and peripheral tissues. Its main role, though, is within the central nervous system. There are 3 receptors that GABA activates, but only 2 will be discussed. GABA is made in cells of the presynaptic neuron from glutamate. The enzyme glutamate decarboxylase uses vitamin B6 in this process. As said, GABA has 2 important receptors, which are GABA A and GABA B. The receptor GABA A being the more important of the 2. It has been found that this receptor is the one that many drugs like benzodiazepine target. GABA A receptor is ionotropic, which increases chloride cell influx when the GABA neurotransmitter is activated. This increase of chloride ions within the cell results in hypopolarization. There is less chance of an action potential. GABA B receptor is metabotropic. It increases postsynaptic flow of potassium and slows presynaptic calcium. This hypopolarizes the postsynaptic cell and prevents an action potential. How does GABA work? Once it is synthesized from glutamate, it is stored within synaptic vesicles within the plasma membrane of the cell. When an action potential reaches the cell membrane, GABA is released into the synaptic cleft. Here it can bind with GABA receptors on the presynaptic neuron. This causes ion channels of this receptor to open, allowing influx of chloride into cell. Chloride ion concentration is highest in the extracellular fluid, and so opening of the channel causes chloride to flow down its gradient. What results is hyperpolarization of the cell membrane, so an action potential cannot fire. GABA then is either reabsorbed back into the cells or it degrades and enters the citric acid cycle. GABA is found throughout the body and is involved in different functions, though its role has not yet been fully understood. It is involved in the function of lungs, intestine, and bladder as some examples. In the lungs, it is part of regulating respiration. There are many medications that act on the GABA A receptor and they seem to enhance the effects of it. Benzodiazepines are an example of a drug that can interact with GABA receptors for either a beneficial or detrimental effect. These drugs exert their effect by binding to the GABA A receptor, causing chloride channels to open faster, thereby leading to increased chloride in the cell. The results are sedation, diminished central nervous system activity, and impaired motor coordination. Benzodiazepines on their own are safe, but when combined with alcohol, the inhibitory effects of GABA can be compounded. This can lead to fatal poisoning. Chloride is important in the role of GABA function, but is also important in the movement of skeletal muscle. The chloride channel is involved in the physiological function of skeletal muscle, and it is needed so muscle can return to its resting state after contraction. There are many types of chloride channels, but the CLC1 is associated with muscle. When cell membrane depolarizes, chloride channels open, allowing chloride ions to enter the cell. Chloride creates a negative state within, making it more negative than what the cell membrane requires for an action potential. This hyperpolarizes the cell, causing skeletal muscle to rest. An example of what could happen if this mechanism was faulty is myotonia. It is characterized by stiffness of muscle due to overexcitability of muscle. Skeletal muscle cannot rest after contraction. Someone with myotonia may not be able to release a pen once it is held. This contraction occurs when chloride channels are genetically altered. Less chloride ions are able to enter the cell, so the plasma membrane becomes overexcited, leading to repetitive contractions even though only one stimulus was received. And that ends the presentation of GABA and chloride channels. Thank you.